Okay, I think we ended with this beautiful slide. Oh, by the way, I'm going to pass this around. Darn it, I forgot to get this. You're going to have your clickers with you on Wednesday, correct? Okay, and if for some reason you don't have a clicker, you will see who? Eileen, you got it. Downstairs, second floor, end of the hall. She's got some that we can use as loaners on the short term. So please do that if you don't have it for Wednesday. I'll let you start with this. Sign in. Then after that, I won't have to be passing around an attendance sheet anymore. Yeah, so we ended with looking at natural selection, and we see natural selection changing in populations, and so I had to show you this gruesome slide of MRSA, because most of you have been attuned to this through your high school programs. Any of you that were in sports, you were told not to share equipment, right? All that stuff. Make sure you washed your gym uniforms and that sort of thing. Because there are strains of Staphylococcus aureus, which are normal skin inhabiting bacteria that can have become resistant to antibiotics. Now, when they're just causing these skin infections, they're pretty ugly, and they're actually one of the bacteria that's responsible for zits on your face and stuff like that. It's not so bad. But if this bacterium should gain access to the interior of your body, it can be life-threatening. So if it gets into the bloodstream, it makes an enzyme that's called coagulase, and when you put this stuff with blood, it just turns into this great big mucus plug. And so you can imagine mucus plugs going through your bloodstream, not a good thing. Okay, they can collect in the heart and the lungs, and that's where they can kill. So, but anyway, if you don't believe, this is what I always say to someone that tries to tell me, well, they don't believe in evolution, then I say, okay, well then, let's treat you with old-fashioned antibiotics and see how you feel about those. Okay, so what we know is that, as we'd said, evolution is one of the best supported theories in all of science. Now remember that scientists use the word theory meaning something that's very well documented and supported. And we'll get into that in a minute about all of that different stuff. And it unifies all of biology together. And it accounts for both the unity and the diversity of life that we observe on this planet. And you will see this quote, "Nothing in biology makes sense except in the light of evolution." That quote has been used over and over and over and over. And we'll keep coming back to this. When you start to see how closely DNA is shared between different organisms, it's absolutely amazing. And then to think that all of these diverse life forms that we have still share so much in common. So evolution is really the theme that brings all of life on Earth together. And we know there's a lot of diversity. Okay, so we know that there are about 1.8 million species. But the theory is that there's many, many more than that, and that we have just not found them yet. And now, as we were mentioning in lab yesterday, talking about the bio-blitz, what did I say about like large animals? How many new large mammals do we find every year? Not many, if any. Okay? So if we only know about 1.8 million species, and estimates of the total diversity on the planet range from anywhere from 10 million to maybe over 200 million, where do you think that is? Where are all those other species? Yeah, William. Bacteria, Archaea, even in the insects. I used to work with a guy over at Kutztown who was an entomologist, and he was in New Guinea. And he was in the Peace Corps in New Guinea, but he was still an entomologist, and he ended up doing his PhD on beetles in New Guinea. And he said, like, every morning I could basically walk out on the path from my house and pick up a new insect that hadn't been described in science before. So it was just amazing. these beautiful colored beetles. And if you come to my office, you have to ask to see the beetles from New Guinea, because a lot of people don't believe me that these things are not painted. They are gorgeous, different colors. But insects. So within the macroscopic, the things that we can actually see without aid of a microscope, the biggest diversity is probably going to be in the insects of things that haven't been discovered yet. Remember, they even found a centipede in Central Park of Manhattan that hadn't been described before. So insect diversity is huge. But really it is what William said, it's the bacteria and the archaea that are really the ones that there's probably a lot there. And so what you're going to find that one of the most, again, to get back to your BioBlitz project, it's while this biodiversity is beautiful and overwhelming, it can be a little bit daunting because we need to assign names and we need to classify these things and try to figure out where they all fit into the scheme of all living things. And there's a branch of biology that names and classifies organisms, and that branch of biology or zoology, anything like that, is called taxonomy. So when you are putting them into groups and assigning them names, that is the science of taxonomy. So what we do is we tend to place organisms into groups, and this is explained also in your handout from the BioBlitz handout, how we will group organisms. So we start out by first placing organisms into these large categories called domains. And then we begin to move down through the groups from domain being the largest to kingdom, phylum, class, order, family, genus, species. Okay, and oh good, they left me something here to write with. Okay, so I always remember them this way. Did anyone ever learn a little, remember I said mnemonics? That's where you start to put together little sentences and little things to remember how to put things in order. Does anyone know the mnemonic? You're looking at me like it's right there. What did you know?
The King Philip came on Friday.
I remember the way I learned it was back in the days when I told you T-Rex was in my yearbook, right? So, but we didn't have domain, but I remembered it. King Philip cried, "Oh, for gosh sake!" But it could be King Philip came on the Friday something. But whatever. That's how I remember it. King Philip cried, "Oh, for gosh sake." Now we have domain, so I have to say, I guess, darn. King Philip cried, "Oh, for gosh sake," or something. But that's how I remember them, just right off the bat. But each time as we move, we are becoming more specific as to what that organism is. So let's look at this. Let's look at this illustration, the Amber Alert. Yeah, Amber Alert. That had me up at 4 o'clock in the morning. How many of you got that too?
Yeah, so like the weather thing.
Yeah, I think this was Amber Alert. Yeah, I have a daughter in college, so I always sleep with the cell phone next to the bed, and at 4 o'clock in the morning, the beep, beep, Amber Alert. So, but anyway, as we look at these organisms, okay, what you do is the domain. Well, let's look at this really cute little black bear. So, we know what a black bear is, but if we see we have the individual black bear, we know that this black bear is Ursa americanus. So that's American black bear. These are the bears that show up in neighborhoods around here every now and then. And they're not particularly dangerous bears. I mean, these are not grizzlies or some of the other, polar bears that have really nasty personalities. I don't want to mess with a bear, but they're not particularly aggressive. So we know the species. The species is Ursa americanus. That's one particular bear. But we have a genus, which is Ursa. Ursus is bears. Okay? So we can have Ursus or Ursus arctica, which is the polar bear. Okay, so there's different kinds of bears, but they're all in the genus Ursus. Then we have a family, and that's the bear family. So when we're looking at the genus of bears, we see black bear, polar bear, brown bear. They're all in there. But now if we start expanding it to family, we can start adding some other kinds of bears in there. We can start adding panda bears and spectacled bears in there. Now, as we move up to the order, we find that they're in the order Carnivora. Okay, what do you think that means? Yes? Carnivores, which means they eat meat. Okay, so they're in the meat eater family. So we have the Carnivora. And now we can start including in that order things like wolves, big cats, big predatory cats. Okay, then all of these carnivores fall into the class Mammalia, meaning that they're mammals. Okay, and why are mammals called mammals? What are they named after? Yes?
No, I was going to say they're called mammals because they don't when they have offspring, they're not in eggs.
They're not in eggs, but some mammals actually do lay an egg. There's a few of them, a few exceptions. Does anyone know what the exception is of a mammal that lays an egg? Yeah. Platypus and another one. Echidna. Okay? But they're named for a particular feature. Yes? Mammary glands. Okay? So they're named for mammary glands. They feed their offspring milk. Okay? So mammals. Then we move up to the phylum, Chordata. That means what? Things that have what? What does the chordata refer to? What cord?
The spinal cord.
Spinal cord. Okay? So they're chordates. They have a spinal cord. They have vertebrae. Then we move up to the kingdom animals, Animalia. And now we can start including a whole lot more in that. And then finally the domain is Eukarya. These are organisms that are composed of eukaryotic cells. So as you see, in taxonomy, we start trying to put things and grouping them. So we start out with huge groups, and then we start narrowing it down as to what it is. So if you're trying to identify something, one of the first things you usually start with is like, okay, well, what kingdom am I going to put them in? Okay, it's an animal. All right, now what kind of an animal? Is it a vertebrate or an invertebrate? Does it have is it chordata or not chordata? Okay, then is it a mammal? Is it not a mammal? So I find this goose out here. What am I going to do? Okay, well, I'm going to look at it, and I'm going to say it's an animal, and it has a spinal cord, so it's a chordata, but it's not a mammal. Okay, it's an avian. It's a bird. It's got feathers. It lays eggs. It doesn't have mammary glands. It doesn't make milk. Okay, so then I start moving it off into a different category. Okay, so this is basically what you do with taxonomy, is trying to look at the characteristics of the organism, move it into these different groups, and then give it a name. And in the past, it was all done on physical features. But now, we have the wonderful science of DNA. And we can start looking at DNA, the genetic material, and start looking at conserved sections of that DNA. And if we start looking at all this variety of life that's on the planet, excuse me there, it's best explained by common ancestry. And the tool of DNA shows us that this is the best explanation for all of this diversity that we see. That you're going to start to see these tree-like structures, and generally what we're talking about is at the base of the tree would be where your ancestor is, and then as you start to move out towards the tips of the branches, you begin to see this breaking off into different groups. So we have the common ancestor, and if the most closely related existing organisms to what they think the first life forms were of course bacteria and archaea. And then as we move up, we begin to see what they call, you may have heard these called lower organisms and higher organisms. And a lot of this just has to do with the complexity of these organisms. But we have prokaryotic and eukaryotic, and we'll come back to all of this stuff later on in this semester. But, you know, most scientists agree that the basic unit of life is going to be the cell, and it's going to be either the eukaryotic or the prokaryotic cell. The eukaryotic cells, of course, have a nucleus, and they have other organelles, which are little structures within the cells that carry out specific functions. And the important thing is there are a few little organelles in the prokaryotes, but they are not bound by membranes. Okay? So the big clue here is internal membrane-bound organelles in the eukaryotic cells, where prokaryotic cells do not have them. But everything that we're going to classify as life is going to have a cell. Okay? And we'll take the life and put it into these three domains: bacteria, archaea, and eukarya. So since we say that the basic unit of life is a cell, now we start looking at the type of cells and the cell structure. So if you actually think about it, that there's only three domains of life, but 2/3 of those domains are prokaryotic. And we start looking about where is this diversity that we don't know what all these organisms are and that we haven't named yet. They're in the prokaryotes. And a lot of that is because we don't, microscopes are relatively new introductions if you think about it in the whole scheme of humanity. I mean, cavemen were identifying different kinds of animals, but as far as like being able to see things that are microscopic, that's really where the world of discovery is going to come in. So there's some sort of a common ancestor that gave rise to bacteria, the archaea, and the eukarya. So both the bacteria and the archaea are prokaryotic. So bacteria you're familiar with on a daily basis. Probably most of you have had strep throat at some time or another, something like that. So bacterial diseases are relatively common, but bacteria are also good. I don't want them to have a bad name. If you love your Activia yogurts and all those things, there's a lot of really good bacteria that we eat. And as I think I'd mentioned in here before, if we took your cells and we separated all the cells that are in and on you and counted them, that you would be way outnumbered by the bacterial cells that are in and on you versus your own human body cells. So, we depend on them. They're good for us. We need them to keep our digestive system working. They keep us healthy. There's a normal what they call flora in and on our bodies. The archaea are the ones that we know the least about of all. The reason we know more about bacteria versus the archaea is the bacteria are very easy to grow in cultures, most of them. So you can, how many of you have been to the doctor and they do the little, stick the thing in the back of your throat to see if you have strep throat, right? The little Q-tips, they stick it back there, they make you gag. Okay, they want you to gag because then they know they got to the right spot. Okay, so they actually do want you to kind of gag. And have you noticed how many swabs do they have? One or two?
That was like one popsicle stick.
Yeah, well, don't they use the Q-tip type things? Okay, they're just looking if they're just using the popsicle stick. If they're going to culture, they're usually going to use 2 cotton swabs. Go back real deep. And one of them they run a quick test on. So they have these little tests that are similar to like the home pregnancy test kits where they can stick it onto a like little matrix and they can get a quick color test. That's what's called the quick strep test. So they can check and see if it's there. But they never want to go off just that. The next thing they want to do is that second cotton swab, they're going to take and swab it over a Petri dish. And you're going to be doing this with bacteria in this semester. They want to swab it and see if it grows. But most bacteria will grow pretty easily in culture. So we've learned a lot about bacteria over the years. The archaea don't grow well on artificial media. And they grow in really wild and strange places, like sea vents on the floor, or of the ocean, or which is where magma comes right up to the surface of the planet, or in hot springs in Yellowstone National Park. You can find some of them growing there. But you know what? They're all over the place here too. But we can't culture them as easily, so we don't know that much about them. And so there's a whole world of discovery that waits with these archaea. And we'll cover later about if we didn't know what we do about archaea, and if we hadn't gotten some of the enzymes from archaea that we have, biotechnology would not be what it is today. I don't know if some of you may have heard of PCR, and we'll cover it later, polymerase chain reaction. That whole process of where you can take one little snippet of DNA and basically photocopy it, I'm going to say, just make copies and copies and copies, which is what they do from crime scenes, why they only need like one little hair out of somebody's head and stuff like that, because they can magnify that DNA, make multiple copies. The only way they can do it is with an enzyme that came from an archaea. So we depend on these things. And so this is a whole science of what's known as bioprospecting, like going into these organisms and trying to find out the kinds of products. I mean, just imagine being able to live at the bottom of the ocean where magma is meeting the surface of the planet and survive in those kinds of conditions. So what kinds of things do they have going on that keep those cells alive? So there's a lot that we can really learn from them. So they're not just this obscure group that, okay, so they're there, they don't make us sick, we're not going to worry about them. Okay, so we can find evidence of these organisms in fossils. So just as I showed you that rock that came from Mars, very similar thing. We are finding that there are modern bacteria and archaea in our fossil rocks. So they've been around. They're unicellular, meaning they exist as single cells. They're prokaryotic, meaning, again, no membrane-bound organelles. And they're incredibly numerous and diverse. And basically they're found on every square centimeter of the Earth's surface, anywhere you can go. You can even find these things in clouds. So they can go way up, sample atmosphere, and get some of these bacteria and archaea from there. So if you see again these trees, and we go, sometimes they're representative of time and sometimes they aren't. But believe it or not, the archaea are considered to be more closely related to the Eukarya than are bacteria when they start looking at DNA sequences. And so as you move along, somewhere a long time ago, bacteria broke off and became kind of their own branch, and then we've got these other groups within the Eukarya. And we'll come back to taxonomy later in the semester. But within the Eukarya, we have 4 different kingdoms. We have the plants, the animals, the fungi, and then what I like to affectionately call the garbage can of the Eukarya, the protists. And I can pretty much guarantee you by the time you guys are closing in on my age, there's going to be more kingdoms than that. Because the protists are basically, if it's not a plant, and it's not an animal, and it's not a fungus, we throw it in the protists. And there's way too much diversity in that kingdom that we call protists for it to be a valid kingdom. We have things in there that are photosynthetic, but swim with a flagellum. So it's kind of like a plant, it's kind of like an animal. But then we've got even giant seaweeds and kelps are thrown in the protists. So amoebas are thrown in there. So we have all of those different things that are thrown into that group known as the protists. But underneath all of this diversity, everything from a single prokaryotic cell that lives on the bottom of the ocean floor to what, of course, because we're really self-centered, what we consider the most highly advanced of all organisms, okay, the human. Underneath it all, we share one thing, DNA. The DNA molecule itself is universal. So, all life as we know it is composed of cells, and every cell contains DNA. And evolution accounts for this similarity that we see, that the fact that there's this unity. So as I say, all cells in all organisms, there's a couple things they keep in common. First of all, that they're a cell. They have cell membranes, they have DNA, and also ribosomes. And we'll come back to the organelle known as ribosomes in a little bit later, but they're basically what interprets the language of the DNA and makes proteins, which just keeps everything functioning. But there's a little bit difference between the ribosomes of prokaryotes and eukaryotes. But this is all in common. The structure of DNA is the same in all cells. It's a double helix. And we'll come back to the DNA structure in later lectures, but let's just say that the double helix of DNA is the same in all of the cells, and that DNA is interpreted or read. So think of it as code that you put into a computer that reads the code, and they make proteins, and that's done the same way in all of the cells. And you think to yourself, how could something as distantly related as an archaea living in a hot spring in Old Faithful in Yellowstone National Park, which is a super volcano, you realize. If that blows, we're all gone. Okay. At least in the United States. Maybe even in the world, another dinosaur type epic event. How could we all be related? But you know what? We are. Okay. And as I say, the basic structure of the cell, the membrane, is the same in all cells. You're going to see that, we'll come back to membrane structure in a little bit, but there are these double layers, these phospholipid bilayers that we'll talk about in a little bit, but the membrane is the same in all cells. So the membranes are the same, the DNA is the same, the ribosomes. While the ribosomes are a little bit different on how they're built, they still do the same job in the cells. And another thing is, there's a universal chemical called ATP. We'll come back to this one later. But this is like currency. Think of this like being your ATM card, okay, or a credit card. You can go and you can use your credit card to pay for anything. Okay, you want a book, you want food, you want shoes, okay, you want medicine. You can use it to get anything. ATP is kind of like the credit card of the cell. You can buy anything with it. Okay, so you can use it to power anything. You can use it to make sugars. You can use it to make proteins. You can use it to, transport things. You can use it for almost anything that you need to pay for. And so you can charge it and recharge, discharge and recharge that chemical. And so all cells use and release energy in the same basic way. Okay. And if you want to know how closely related different organisms are, this one is just so cool. Scientists can do some really cool things with DNA now. What they did here is they took this was actually, I think this is done in Korea. They love to do this kind of stuff in South Korea. They like to do these little genetic things. They took little kitties, and in these little kitties, they took the protein from jellyfish that can make them glow. Now, how many of you have been to Camden Aquarium, or what do they call it now, Adventure Aquarium or whatever, and they have that really cool jellyfish exhibit with the black light on them that has a UV light, and the jellyfish glow. So they have this protein that can glow under ultraviolet light. So they took that protein, the genes that code for the protein that makes that glow protein, and they inserted it into kitties. And so it's in the kitty's bloodstream. So where, of course, kitties we know are covered in fur, but there's certain areas of the kitty that has less fur on it than others, and that's like the ears and the nose and that sort of stuff. And so what they did is they put the protein for that glowing jellyfish protein into the kitties, and then stuck the kitties under a UV light. And we have glow-in-the-dark kitty cats. Okay? So that's a jellyfish protein put into a mammal, and it still functions. can still be read. Okay? So I guess you could do this and then you don't need a night light, right? You just have your kitty get up and, but no, you have to have a black light to see that. But other things, I mean, just think about the fact that, you know, we can get human insulin now made. Humulin is a human DNA sequence that was put into bacteria. The bacteria make the human insulin, which they then harvest from a fermentation tank, and then you get injections. So we can have bacteria making human genes. We can take jellyfish genes and put them into kitty cats. So we can do all kinds of really interesting things with DNA. Okay, so now we're going to go to the scientific method. I think I already had this PowerPoint online. I'm not sure if I did or not. I started thinking about it at 4 in the morning when the Amber Alert woke me up, whether I actually have this one online ahead of time or not. But let's start looking at practicing science and how scientists approach a problem. And what I want you to think of in this class, and I've said this over again, and I mentioned it yesterday when I did mention it in all the different lab sections when we talked about the textbook, and a lot of you being really overwhelmed with how much information is in that textbook. That is a textbook that is going to last you through your entire college career for biology. There's a lot of detailed information in there. I don't want you focusing on individual little facts. Instead, I want you to focus on big concepts. Science is really not a giant collection of facts to be memorized. You may need to memorize vocabulary. I said that from the beginning, and I'm sorry, there's really no other way to get around it. When you learn a new language and think of biology as being a new language, you have to know the words. And some of it does come to memorization, and they'll become second nature. But as far as the other parts of science goes, it's really not just trying to memorize facts. It's really more concepts. Understanding concepts and understanding that it's a process and understanding how all of these things relate to one another. So science is this process, and it's really a method. So when we're looking at scientific investigation, what we're trying to do is explain the natural world. Science cannot explain the supernatural. Science cannot get into religion. Okay, it can only get into the natural world. Only the things that we can see and touch around us can science explain. So what science attempts to do is to explain the natural world, and it's done through this process. Generally, people will make an observation. They'll see something, they'll question that. They're going to propose some sort of a hypothesis as an explanation for what is being observed. Then they want to actually devise tests of their hypothesis, and then they start discarding ideas that don't fit with that hypothesis. And what do we mean by hypothesis? Well, we're looking at something that is a proposed explanation. And a good hypothesis must be testable and potentially falsifiable. And you're going like, what? Okay. The important thing is with scientists, it's a little nuanced difference, but it's a really, really important difference. Scientists don't attempt to prove something is correct. They attempt to prove that it isn't something. Okay? Now, I can't prove, it's just like the thing, innocent till proven guilty. How do you prove somebody's innocent? How do I prove you didn't do something, that you did something? Well, but I can prove you didn't. Okay? So that's what science attempts to do. They try to falsify things because there could be other explanations for things. So for us to be able to actually run an experiment on something, we have to come up with a hypothesis that is testable and one that is potentially falsifiable, that we could potentially get a negative result. I'm not saying we would, but it's possible, the way that experiment is designed, that we could say that it's false. Now, an important difference that the general public doesn't understand about scientists. Scientists feel very misunderstood by the general public, okay? Particularly in today's media, where, you know, anything can be put out on the out on Facebook and the national news and all this kind of stuff, and things can be taken out of context. But there's a huge difference between a scientific theory and a hypothesis. Theories are powerful, broad explanations that have been explained by a large number and sets of observations. It's very, very well supported. There have been very many tests that back up that hypothesis. So, again, you hear all the time, evolution is only a theory. So, to the general public, that means, oh, well, you know, it's not very well supported. Well, in scientific community, a theory is a big deal. It is very well supported. Okay? I mean, I can take your gene for insulin and put it in a bacterium and have it make your insulin for you. I can take a glow-in-the-dark kitty cat, you know, make a glow-in-the-dark kitty cat. I can do all these kinds of things, okay? This shows, you know, evolution can be demonstrated through antibiotic-resistant, you know, look at MRSA. That we have many, many, many examples that support that hypothesis. Support it, and it's now become a theory, okay? So, theory is a big deal. It's not just a word that scientists throw around out there. Okay? Now, if we want to go through and test hypothesis, so we're coming up with, and this is basically what science does. Scientists move forward. We advance science through the process of hypotheses. So hypotheses is plural, hypothesis is singular. So there's two different ways to approach a problem. Inductive reasoning and deductive reasoning. Now, in inductive reasoning, what you're doing is combining a series of general observations to try to explain something. So, you induce it. Let's say if I'm looking at vitamin C. A lot of people think that vitamin C helps to prevent the common cold. Well, vitamin C is found in fresh fruits and vegetables, and vitamin C has been shown to be an anti-inflammatory. And what gets inflamed when you have a cold? Your nose and your sinuses, and all that sort of stuff. So this has been through mainly observational types of things like this. It's inductive reasoning. Okay, well, fruits and vegetables are high in vitamin C. People that eat fruits and vegetables are healthier than people that don't eat fruits and vegetables, and they have that in it. So that you make this kind of inductive reasoning that vitamin C must be good for the common cold. And they even have tons of products out there. One of them named Emergency. Have you seen that product on the market for colds and stuff like that? So that would be inductive reasoning. Deductive reasoning is one where you can set up an experiment and do what we call if-then statements. Okay? So this would be in deductive. If vitamin C reduces the severity of colds, then people who take vitamin C should have fewer colds than those that don't. Okay? So it's something then that would be deductive reasoning. Okay? So here we go. I didn't realize I had this one in here. Okay, so the hypothesis: consuming vitamin C decreases the risk of catching a cold. Fruits and vegetables are high in vitamin C. People who eat fruits and veggies are healthier than those who don't. It's an anti-inflammatory. That's inductive reasoning. Okay, so we said deduction usually takes the form of predictions based on experimental results, if-then statements, and you set up experiments to test those types of things. So the process, again, observation leads to questions. You formulate a formal hypothesis that then is testable and potentially falsifiable. You make a prediction, and then you run your test. If the results do not support the hypothesis, then you either revise your hypothesis or propose a new one. But there's always the possibility that it will support. And your prediction comes there. Now we say it supports the hypothesis. And we make additional predictions. Okay, scientists, you talk about, like the P word. Okay, they're very careful about using this. Scientists generally will not say they've proven something. They'll say their hypothesis is supported, because there could potentially be another explanation. But they can say they proved their hypothesis false, but they don't say that they proved their hypothesis. So I'll be looking for that in any of your writing. We proved ourselves right. Well, you just supported your hypothesis. You didn't necessarily prove you were right. Okay. So that's another thing the general public will hop on scientists for their language. Well, they didn't say they proved it. Well, scientists don't say they do. Now, mathematicians, they have proofs. They prove things. Okay? But generally not biologists. Okay? So a hypothesis that fails the test is rejected and is considered disproven. But a hypothesis that passes is supported, not proven. Again, why not? There could be an alternative explanation for what we saw. So how do we go through this? Through experimentation. So scientists' experiments are extremely important. They're contrived situations. And we'll go through this in lab. You guys are going to be doing experiments. You're going to be setting things up. You're going to be doing your own experiments in there. And it's really important that you understand what you're trying to do in an experiment is set up things with variables. So factors that can change. Okay, so you want to have different scenarios. You want to have different factors that will change in value under different conditions. And the two basic types of variables that you're going to encounter in experimentation are what are known as independent and dependent variables. The independent variables are those that you can manipulate. So I can decide whether I'm going to give you vitamin C or I'm not going to give you vitamin C. If I'm going to give this group C and that group D. Okay, I can control that. The dependent variable is then what I'm going to measure. Okay, so I'm going to measure how miserable you are if you have a cold. Yes?
Would like human error be considered an independent variable?
You
mean like human error in what kind of?
I mean, say like you were supposed to measure out, a certain amount of, milligrams of some kind of, substance, and you were probably off.
That would be experimental error.
Experimental error.
That's a different thing. It's not considered to be part of the variable of the experiment. That would instead be just in what they call error. So in how you carried out the experiment. It wouldn't be something you'd build into your experiment. So this would be in your design. So you'd have your independent, variable, which would be what you're designing the experiment for. So this just comes up in the design, but that's actually in carrying out the experiment. Okay. So independent is what you're setting up that you're going to have different from one to the other. The dependent variable is then what you're actually measuring. Okay. Okay. And we always want to do what we call controlled experiments. We want to have control over the variables. A really good experiment is going to test a single variable. We don't want to start getting into a whole bunch of, if you start getting into complex experiments. Okay, so I want to see if vitamin C and grape juice and salads affect colds. So I'm going to give this group, I'm going to give part of this group grape juice and some of them vitamin C, and then this group over here is going to have salads and grape juice, and this group's going to have this, and I start comparing all this stuff together. That becomes extremely difficult to sort out the data. A really well-planned, strong experiment is going to have only one thing different between the two. And we usually want to have what are known as experimental controls. Those are things that maybe don't get the treatment. so that I know what it is. So for a well-designed experiment, if I wanted to see if vitamin C was going to help prevent colds, I would take everybody in this room and I'd split them randomly in half, and you guys would be a great experimental unit because you're all about the same age. Okay, and so I don't have a lot of variability. I don't have any geezers in here. Okay, I don't have any little babies in here. I've got young adults in this room. So I'd get you, put half of you. I'd make a mix so that I have half male, half female in each of my two groups. One group I'd give vitamin C. The other group I wouldn't give vitamin C. And then I'd come along and blow some rhinovirus up your noses. Okay, inoculate you with a cold. And I would see, did my people that get vitamin C, when they were inoculated with the cold virus, did I see fewer colds in people that had vitamin C versus those that didn't? That's a good experiment. You might not think so when you're in the group that got the rhinovirus, but anyway, that would be a good experiment. I'm not putting a whole bunch of things, like what if I put all the guys in one group and all the girls in another group, and I gave all the girls vitamin C and none of the guys vitamin C, and then I inoculated, then how do you know maybe it's not a difference due to being male or female? I've complicated things. So we want experiments to have just one variable in them. So I'm going to give you a scenario here also about the importance of having an experimental control. Okay, so imagine this now. You are on a research expedition to a remote location. You're going somewhere where no one's going to be able to get to you. So let's just say Antarctica. So you're going to Antarctica in the middle of our summer, their winter, and you go down there and you get snowed in. And they can't come down there and they can't get you out. Okay? And you're in this group and you don't have a physician with you. But you do have a little medical computer and you've got some medicines. And 3 members of your team come down with 3 different illnesses. And what you can do with this little medical computer is you can put in the symptoms, and it'll make a little diagnosis, and then it'll tell you a medicine that you may have in your kit that could be used to treat that condition. So luckily, some of the drugs that have been used to treat the conditions in the past are available. And this is the information that the computer gives you. Okay, so you have 3 different people, and I'm just using letters here. So, the first individual, number A, letter A, has disease A. And the computer tells you there's a drug in there that I'm going to say A because it matches with that individual. And individuals treated with that medicine have a 23% survival rate. We have another individual that has disease B, and there's a drug that matches that disease. Okay? And if they're treated, they have a 56% survival rate. Those with C, if treated with drug C, have an 89% survival rate. Okay? Who are you going to treat?
C. No, A. Definitely.
Why can't you treat them all?
Yeah.
Okay. One possibility is treat them all. And you're saying,
Why can't you treat them all? Is there not enough, like...
no, I mean, I'm just deciding, which ones would you treat and why? So you're just saying, if the medicine's there, treat them all.
Yeah, absolutely.
Okay? You said C. Why did you say C?
Because C has the highest chance of surviving.
That's the highest chance of survival is to treat with C. Okay?
I'd see like each one what their survival rate is before you give it to them, like before they get the medicine.
Well, before they're already sick.
I know,
But what you're I like what you're saying, I just want you to say it in a different way.
like if you don't treat C, like if the survival rate is already high, you don't need to treat it.
So what you're saying, what's missing from this scenario? You've just said it. You said it. I just hoping you're saying that I can get you to come out with the,
Survival rate before the treatment.
not before, but without. That's what we're missing. How do I know what it would be if I didn't treat them? Now, does this change who you'd give medicine to?
Yes.
Exactly. All right? What happens to A? Only 23% survival rate if treated with that medicine. But guess what? If he's not treated with the medicine, his chance of survival is only 10%. So guess what? I'm pulling that one out and I'm giving that pill to them. Okay? Now, with B, that is such a small difference. It probably doesn't make a heck of a lot of difference whether that person is treated or not. There's a little bit of chance that it may help. If I've got the medicine and I'm not worried about using it up for somebody else that might get sick later, then I might use it. It would depend on the circumstance, but probably it's not going to make that much difference one way or the other. Now, how about C, which appeared on the surface to be the no-brainer? What happened? We actually...
The medicine is not a very good. If anything, it's actually bad. It will backfire.
Yeah, it's harmful. It's apparently harmful. So see what happens when you don't have a control. Okay? So this is what's extremely important in a well-designed experiment. What happens if you do nothing? Okay, so which is why, how many of you know what's in the Hippocratic Oath, which doctors take? You know, they have to, they do that from, it comes from Hippocrates, and what is it?
Well, I know Hippocrates
Mm-hmm.
I think it's if they end up a patient gets off their hypotheses,
Mm-hmm.
and they have to
Drink oak serum. Okay, but what else? What's the first rule there? Yes.
not cause them harm.
Yes. The thing is, the first rule of the doctors is first, do no harm. That's the number one rule of the Hippocratic Oath. First, do no harm. You don't know, if you don't have controls, what that could be. You might think to yourself, I am doing this guy a huge favor. 89% rate of survival if I give him this medicine. But guess what? You've just reduced his chances of survival by 6%. So we see the importance of a well-designed experiment and experimental controls. Okay, so we will pick up here on Wednesday. And what are you all going to have in your hand in lecture on Wednesday? Clicker. And what are you going to have in your hand on Thursday? A lab notebook. Okay, have a great weekend. Feel free to email me if I haven't done something you think I should have done by whenever you think I should have done it.